question. This is going on to question 3. So we know now we're on a new part of the syllabus. So we've moved off from that last question. There's no intro to this question. You will get some questions that are like that. There's no information at the top. It just goes straight in with what is an enzyme. And it's 2 marks for this, so you need 2 points. So your first point needs to be that it is a catalyst. So it speeds up chemical reactions, or speeds up a chemical reaction. That would get you 1 mark. Then we need something else for that extra point. So we need to be thinking about that enzyme is not part of the reaction. It's not used up in the reaction. So an enzyme is not used up in the reaction. And that gets you that extra mark. Now remember, with those questions, what we're looking for is 2 points. You're not looking for the same point repeated, so 2 different facts. 3b. Enzymes are sometimes used in the home and in industry. So you're looking at application of enzyme knowledge now. So draw 1 line from each enzyme to the correct use. So we need to match up one of these to each of these. Now, when you read through these, you need to see if you can try and guess, or if you know any first. So you're looking to see if there are any that match up straight away. Now, one of the good ones is here is check out the names. That one's got protease. And if you look, one of those is about breaking up protein. So we know straight away that that one must match up with there. So if you've forgotten that knowledge, use whatever you can to help. So protease breaks down proteins. Lipase, that breaks down fats. So when we're looking for something that talks about breaking down of fats. And the only one we've got is removes grease stains from clothes. So we're matching up to there. Again with this, sometimes these questions become a mess because you can't see clearly where the line is going, or you make a mistake. If you make a mistake, make sure that you're showing that that one's wrong. Okay? Sir, could we just get Amy moved towards the back, please? And our final one here is isomerase. So what we need to do is to try and work out, okay? You should know that that will break down sugars, okay? If you didn't know that, these are all talking about foods. Protein, grease stains, match it up. And we've got here that we've got glucose syrup and fructose syrup. So our last line goes to here, okay? So on some of it, you're making educated guesses. If it's saying match them all up, don't leave it unmatched. Even if you do have to guess, you will need to do that. You might get that point. Okay, question 3C. Fructose and glucose are 2 types of sugars. Fructose tastes much sweeter than glucose. This means that a smaller amount of fructose can be used to give the same sweetness. So what is an advantage to use fructose instead of glucose in slimming foods? Okay, it's basically told you the answer. You probably would be able to have a guess at this if you didn't know this knowledge. So you need to be putting that there's either less energy or fewer calories in the fructose. Okay, and it has to be fewer calories or less energy. You can't put it's less fattening. You're talking about fats there, not sugars. Okay. Now, building this question up, remember they start off asking a few easier questions first while they're building up this. Then they might get more difficult to the end. Remember, if you're struggling and on the end questions you think, oh, I can't do this, I'm not going to be able to do anything else in the paper, you can, because your next question will go back to a few easier ones again. So try every question. So this time we're looking at figure 3, and it shows how an enzyme can be used to change glucose into fructose. The enzyme molecules are firmly attached to beads of gel inside a glass tube. So we've got our glass tube, we have our beads inside, which have the enzyme attached. Okay, our glucose goes in and we get fructose going out. So these enzymes breaking the glucose up so that we get fructose. And we're giving 2 advantages again, so we're ticking 2 boxes. 2 advantages of attaching the enzyme to beads of gel. So again, we need to look at all of the answers that we've got before we decide, and read through each one and either cross it out that you know it's completely wrong, or you need to put a question mark or leave it there if you're not sure, and a tick if you know it's definitely right. So we're looking at advantages of attaching the enzyme to the beads of gel. Nothing else, just those advantages. So the enzyme can be used for more than one type of reaction. We know that's wrong. Okay? Enzymes can only be used for one. It's used for this reaction. The enzyme can easily be used again. That could be right. The enzyme would be denatured. Well, there's nothing in that question that talks about a change in temperature or a change in pH, so that wouldn't happen. So we know that's wrong. The fructose solution produced does not have any enzyme in it. Okay, well, if those enzymes are attached to the bead, when that fructose comes off, that is possible. Okay, that we wouldn't have that on there. And there is less contact between the enzyme and the glucose. Well, the glucose is coming through. The glucose is going to be in contact with that, so we know that's wrong. And that leaves us with our 2 correct answers. The enzyme can be easily used again, and the fructose solution produced does not have any enzymes in it.